Thank you for giving me again the opportunity to moderate this next panel. It's on developing GHF 201 for clinical trials, and I'm happy to introduce the speakers. Dr. Or Kaklon, he and I, we have the same hairstylist, and then we have Dr. Alexander Lossos and Janif Glaser. Is that how you pronounce your last name, Dr. Glaser?
Yaniv, that's fine.
Yaniv, okay. Dr. Kaklon is assistant professor in the Department of Neurology at the Hadassah Hebrew University Medical Center. Dr. Lossos is professor of neurology and head of the Leslie and Michael Geffen Center for Neuro-Oncology at Hadassah Hebrew University Medical Center. And Yaniv Glaser is the chief executive officer at GHF, the company that's commercializing GHF 201. So I think this is a very nice combination of people who are leading a compound from the foundational research aspect towards the clinic. And I'm very happy to hear from all three of you. I believe, Dr. Kaklon, you're going to start.
Great. Okay. So I'm just going to give you a brief intro. Actually, it was some of it was given by Dr. Minassian. Actually, most of it. So we have glycogen as a. Can you see my cursor, by the way? No, you can't see my cursor.
Your cursor. Can you move it again,
Yeah.
Dr. Kaklon?
Can you see it?
No, we can't actually.
Okay, never mind. Okay, so on the left-hand side, you can see. Sorry, on the right-hand side, you can see a normal glycogen molecule. It is globular and it is also it can dissolve in the milieu of the cell, in the in the hydrophilic milieu of the cell, because it is actually the edges are actually hydrophilic, and because of the branching. Okay, so glycogen molecule is generated, synthesized by elongation or adding glucose residues one to another, and then every 6 or 7 residues there is a branching effect, and if you iterate this process, you end up with a globular molecule which is soluble. However, when the elongation is not balanced by enough branching effects, you get this elongated molecule called polyglucosan, where, as you can see, the hydrophilic edge, the hydrophilic edges are not on the surface of the molecule, so it actually excludes a water molecule, precipitate, and generates a structure which is more like starch than glycogen, which can precipitate it and clog axons, and this is the cause of the disease. And as I said, Dr. Minassian actually introduced it previously. Next. So the therapeutic approach actually could be two-faceted. It could be either changing the ratio, the activity ratio between the synthesis and the branching. Okay, so the less synthesing and more branching there is, the more the balance between glycogen and polyglucosan will be tilted towards glycogen. Okay, and the other approach is just to degrade either polyglucosan or both forms of glycogen and polyglucosan, and both approaches will actually reduce the pathogenic polyglucosan and are supposed to ameliorate APBD symptoms. Next. So this disease was APBD, was described in the 80s. The mutation was discovered by Dr. Lossos in the 90s. And actually, this is just some clinical observations that we see the accumulation of polyglucosan bodies in glia and the neurons, and the white matter abnormalities. You see here in the dashed circles with the pyramidal signs that also were mentioned previously. Next. So it's a leukodystrophy. And what is interesting, can you actually click on this black rectangle? to activate the movie? Thanks. So I just wanted to describe in brief the approach we took in order to discover GHF201, which is the subject of this talk. So we actually had a hypothesis-free high throughput screening campaign in which the output, what we looked for, actually more the readout, was just molecules that can reduce polyglucosan staining in skin cells, in fibroblasts derived from skin cells of patients, okay? So the readout was very simple. Since you can stain them with periodic acid Schiff reagent, whatever molecule can reduce this staining is a good candidate. So we just noticed that we focused on the end result, not on the mechanism. And then we discovered, we screened several libraries. One of them was 11,000 compound library. We discovered some hits. The next stage was to decipher the mechanism of action. As you see here, we had some interactome, some candidates, and the mechanism of action was also discovered by discovering the target, which is LAMP1, and we saw that it actually enhances autophagy. There, next, we may ensure that also at the histopathological level, the compound was able to reduce polyglucosan in different tissues to where it was biodistributed in a APBD mouse model. Then we confirmed, as you can see here, that this film is a bit it's stuck a bit, but you can see that actually the treated animals moved much faster than the untreated animals, and this is just one parameter which was modified among the many others of the health of the animal. And once we've confirmed this and published this, the way was paved towards clinical trials. And next, I think that's where I'm. Yes, okay. So initially the compound was given to the mice as a DMSO solution, and we were limited by two things. First of all, the solubility of the compound, because we couldn't use levels which were too low of DMSO, because otherwise the compound won't dissolve. On the other hand, we cannot inject to mice and of course to humans to high levels of DMSO. And therefore, we actually joined forces with Dr. Nissim Garti and his company, Latropic Delivery Systems. And then we actually formulated he formulated the compound GHF201 into a special tailored nanodroplet where the compound was on the interface and actually enters the body through the jejunum, bypassing portal circulation, therefore portal metabolization. And as you can see from this pharmacokinetic profile, the biodistribution to all organs was increased. Number one. Number two, it can be also available orally. So we thought this would be also the right excipient to give to the patients. So next. And then actually we started with the clinical study, and which was compassionate use. And now I pass the baton to Dr. Lossos.
Yes, okay. Hello to everybody. So I will shortly speak about the clinical implication of the substance, which we call substance A or compound A. Next slide, please. Yes, okay. Next slide, please. Yes, so our purpose was to see if the substance A may be implicated in treating people with the APBD. Now you have to understand, everybody understands that APBD, since it is a genetic disease, so people accumulate polyglucosan bodies since their childhood, their birth. And over the time, polyglucosan bodies derange the function of neurons, in different mechanisms. One of the mechanisms is just to cause abnormal axonal transport, and that's why what we saw on one of the previous slides of Dr. Kahlon, a prolonged neuron which was blocked by the polyglucosan body, so it cannot function. This was the purpose of our program. Now, the process of our program was a formulation of a program for treatment on a compassionate basis. That's how it was approved by the Ministry of Health in Israel, and it was approved also by the local IRB as a compassionate treatment. So it is not a trial, it is not a study, it is a compassionate program approved for only 3 people. That is all that it is. Now, the recruitment program, the recruitment process was very strict. It was according to the strict inclusion and exclusion criteria, and it was controlled, organized, and performed by an independent researcher appointed by the local IRB. That is all to it. For example, just one of the inclusion and exclusion criteria, the main inclusion criteria was a patient who is a heterozygote, a homozygote for the common mutation. So a heterozygote person could not be recruited to this program. This is one example. So what our outcome was is to see the safety, first of all, the safety of the treatment, and to see if we see any indication of its beneficial effect. The safety is very strictly observed, and we not only evaluate patients every certain period of time, but we also see all of their blood tests and ECG and so on. Go on to the next, please. Yes, thank you. So what are the criteria that we use to observe if the treatment is effective in some way or other? So first of all, you see the patient's MRC scale. MRC scale is the evaluation of the motor, of the muscle strength of people. In this case, in the legs, APBD affects primarily legs because of the length of the neurons in the spinal cord and in the periphery. So we see that there was a indication of improvement of the MRC scale. You don't see my arrow?
No, we're not able to see your cursor.
Okay. So what we have observed is that there is a kind of improvement of the MRC scale of the motor strength of the leg muscles. Now, in the first patient is already about 30 months on the treatment. There are 2 periods in her situation. The first period was that there was quite an observable improvement of the strength of the muscles in the legs. Then she fell down and she broke her ankle. She had an operation, and thereafter she had a rehabilitation, prolonged period of rehabilitation, and during this period she has a worsening of the strength. Currently, she is after the rehabilitation period, and she again becomes a little bit stronger. The second patient, we see an observable improvement in her leg function, And the third patient, we see an initial indication of at least few muscle groups that are improving. Now, this is the clinical observation. The second observation that we have is on the fibroblast. We take fibroblast to evaluate the amount or the sum of the polyglucosan bodies in their fibroblast. So on the first column, you see fibroblast before initiating treatment, and polyglucosans are red. You see them as red. On the first time point, so this is the second biopsy of the skin fibroblast, cultured skin fibroblast, you see that we do not see any observable polyglucosan bodies. So they have been cleared. And on the second time point as well, on all three patients, we see that the polyglucosan bodies that were clearly observable before treatment, they disappear. So we don't see it. It is a biological effect that we see on a cellular level. So it is something that cannot be argued with. We also saw improve on the rightest column. You see the calculated amount of polyglucosan bodies in the fibroblast. These are so all three patients had at least 3 skin biopsies, 1 before treatment and 2 during the treatment. Next slide, please. Okay, we had also a kind of indication of the effect of the substance A treatment, testing the neurofilament light chain levels in plasma over the time, which currently are accepted as a kind of indication of neurodegeneration. So we see that the neurofilament levels become less and lower and lower over the time. These are the clinical and biological indications that we see. Do we have another slide? No, okay. This
You
is
want me to
for
take this?
Dr. Kahlon.
Yes,
Now,
I will take it.
another thing that I would like to add is that testing the standard blood test like CBC, biochemistry, ECG, urine, coagulation tests, we saw no side effects, no adverse effects of this treatment on patients. They all told us that they feel well and they feel no adverse effects, and that is all. Next slide for Dr. Kahlon. Thank you.
Yes, I actually briefly mentioned this in my previous talk, but I just want to point out 3 observations. So this is just the PCA profiling, principal component analysis of the dimension reduction of a panel of about 200 metabolites. And what you can see here is in red, you can see the healthy control, in green, in blue, the patient, and in green, the treated patient. So what you can see here is actually that after the treatment, the center of gravity of this ellipsoid gets closer to the healthy controls, which means that in general, the metabolomic profile, the metabolite composition in the blood is improving or gets closer to the healthy control. And here you can see actually the different metabolites that we analyzed, and you can see here 3 kinds of effects. So here you can see the healthy control. Here you can see the patient. As I said in the previous talk, in general, these are mostly lipids, phosphatidylcholines, and the fatty acids, for instance. And in general, this is also known from the literature. There is a decline in the levels of lipids in the blood because the patient utilizes, metabolize lipids instead of glycogen for energy, because glycogen is relatively unusable as a source of carbohydrate energy. And what you can see, so you can compare the patient to the healthy control. And as you can see here, with the lipids also after the treatment, the lipid profile in the blood gets closer to healthy control, which we can interpret to say that there is less use of fatty acid. Sometimes there's even overshoot. Okay, so the direction is to increase the levels of metabolites, but it's even more than healthy control. And we also have to say that for some metabolites we get an antagonistic effect. So if it's increased in the or in this case if it's decreased in the healthy control, it's actually increased in the treated patient. Okay, and each profile was asked about precision medicine. For each patient, of course, at each time of treatment, the profile might be a little bit different. And of course, we can learn a lot about the special treatment and how to personalize the treatment from this kind of holistic analysis. Next, I think it's not me. Yes, so this is an example of a patient who was treated for less time, okay, and as you can see here, there's more of antagonistic effect, for instance, and while you can see also that overall the treatment brings the metabolomic signature closer to healthy control, the effect is less pronounced as compared to the patient who was treated for a longer period of time. Next. So I think it's you, Alex, you can describe this. You're muted, Alex.
Yes, we can see videos of patients. This is currently, and we of course obtained their informed consent for these videos. And the videos cannot be used anywhere else.
And they were paralyzed before, right? I think we don't have a before picture.
So you see when every patient started the treatment. Go on to the next, please. You may go to another to the third patient. Thank you. Thank you very much.
Okay, so thank you, Dr. Kahlon and Professor Lossos. So basically, after reviewing the background of GHF-201 in terms of the basic science and the clinical data from the 3 patients, the goal of GHF in APBD is really to transform this into a drug development program, and subsequently to submit these findings to the relevant authorities for approval. Now, to accomplish this, it is essential that the process is conducted properly, cost-effectively, and aligned with patient needs. Well, basically, obviously, unlike this cartoon. Now, this was and is the company's objective as well as its commitment to the APBD community since its inception. So how do we plan to achieve this? Now, since the audience, as I can see in this conference, is diverse, I would like to briefly outline the necessary stages in drug development, because I'm sometimes getting questions from patients about this. So first step is basic research, where molecules are screened based on various criteria and various insights to identify potential hits for disease model. Unfortunately, there is no guarantee that such effort will eventually yield positive outcomes, as it is frequently seen in other projects in the healthcare space. The second stage is the preclinical phase, focused primarily on toxicology studies, both short-term and long-term, typically involving cellular level tissue as well as animal models, in most cases of 2 species. These 2 stages usually take about 3 to 6 years, but could also be even more. Once it is established, then the drug is, I would say, reasonably safe, and the benefit outweighs the risks. We can then move to the clinical phase, which involves human trials. And by the way, preclinical studies often continue in parallel with the clinical trials. So the clinical step consists of 3 phases. Phase 1 is conducted in healthy volunteers to assess the drug's safety in human. Phase 2 and 3 involves patients examining both efficacy and safety over time, starting first with small groups, then with larger groups. And these stages can take years to complete. And many times, due to emerging data, there is a need to repeat steps or conduct additional studies. Now, after completing these stages, the data are obviously submitted to the authorities for review, and hopefully with the approval, which could also be a lengthy process in itself, something like 1 or 2 years. So the whole, the time that it takes from the starting the development till you get to the market can be a lengthy program, which goes up to 15 years or so. But this is an example for a traditional drug development process. However, our development pathway is different, as we are targeting rare disease. This gives us several opportunities to expedite the process. First of all, there is a pressing need for solutions of life-threatening diseases without treatment, where patients continue to deteriorate, creating a sense of urgency. Now, regulatory bodies are also aware of this, and like the FDA, they allow faster approval pathways for such drugs if meeting some strict criteria. Since rare diseases typically involve small patient population, clinical trials are smaller in scale, involving fewer participants, and have a shorter follow-up terms. Now, all these factors, combined with tax incentives and fee waivers for companies like us developing such treatments, that may reduce the financial burden. Now, in certain cases, when safety risks are minimal, there is even a possibility of conditional approval, if not all criteria are fully met, but this is very, very rare cases. Now, the next slide illustrates the timeline differences between the drug development for rare diseases versus conventional development, thanks to the factors that I just outlined. So, as I said previously, it usually takes 15 years or even more to develop a regular drug. But as you can see, for orphan diseases, it can get us to the market within less than 10 years. In our specific case, we are currently in the preclinical stage, which I'll discuss shortly. Our goal is to develop the treatment for APBD within a time frame that I would say falls at the shorter end of the average orphan drug development timeline. Now, as mentioned previously, the preclinical stage is complex, and can take years. However, in our case, the fact that we already have clinical data from 3 patients, even as Professor Losso said, it's a small number of patients, it's not a clinical trial setting, it still has significant weight in planning this phase. This will be, I would say, advantageous as we present the preclinical results, which are promising so far before advancing to the clinical phase. Another critical, I would say, another critical aspect of drug development is the production of the drug itself. Now, this stage of synthesizing the API, or active pharmaceutical ingredient, or the drug substance, through the development of the final product, which is called also drug product, could be also very complex. For example, we must ensure that there is a consistency of the product, whether produced in small scale or large scale, and meeting the what's called the GMP, or the good manufacturing practices, until you reach until you reach the point where you start the human trials. We are in an advanced stages to ensure that this will not be a limiting factor as we enter the clinical phase. And here you can see the example of how our drug substance look like and the drug product. Now, in the next couple of years, our focus as a company will be pushing the development forward from preclinical stage to the clinical trials, while ensuring GMP production of the drug substance and drug product. This is an important shift of what we do so far from the academy to work according to the FDA regulations. We will leverage all available regulatory opportunities to advance the process in the most efficient way. And at the same time, we are committed to strengthening the IP of the company, as well as expanding both organically and in terms of activities to exploring potential collaborations, particularly for additional indications. Now, from the beginning, I've shared with you the differences between the development of a regular drug versus an orphan drug. Beyond, I would say, the goodwill of everyone involved in and the positive results so far, I think nobody will be surprised that financial resources are definitely required to support these efforts. Now, just to give you in context, the development cost of standard drug can reach hundreds of millions, sometimes even more, of dollars, while orphan drug development costs tend to be lower, and that the exact figure really depends on the disease urgency, whether there are approved treatments, and obviously based on the outcomes. In our case, we are working to advance our activities cost effectively and while adhering closely to regulatory guidelines, especially those of the FDA. We are currently in round A of funding, which intended to carry us through the completion of phase 1 and preparation for the pivotal trial. We've already secured a significant portion of this round, and I would like to take the stage and encourage anyone who can help connect us with suitable investors, both for this round and beyond, to reach out. I would say that this is the time. Can you move to the next slide? Because I think I've lost control. My last slide. Next. 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 Great, thanks. So I've mentioned that we are a team made up of, I would say, incredible individuals, experts in their field, starting with our research partners, Dr. Or Kahlon and Professor Miguel Weil, who researched and discovered the molecules that currently make up the company's portfolio. Professor Lossos, well known for his work in the APBD field and beyond, together with Ms. Anat Mordechai, who is the coordinator in the compassionate use program. Professor Garty, the owner of LDS, which developed for us the liquid formulation to be given orally to patients, is also recognized and a well-respected figure in his field, both locally and globally. And of course, last but not least, the GHF team, led by the Schneider family, Shlomi and Adi, who I would say with their spirit of entrepreneurship and boundless optimism, worked together with other patient families to promote the founding of the company and the development of treatment options such as GHF-201 for all APPD patients worldwide. Thank you.
Thank you to all the panelists who talked in this session. Unfortunately, we're a little bit short on time right now, so there were a few questions in the chat, some of them, or in the Q&A, some of them were actually very focused questions, and I would like to ask you, Dr. Kaklon, Dr. Lossos, and Yaniv, can you maybe look into the Q&A and answer the specific questions just right there? I think from when I just kind of think about what from the patient perspective here in the US, for example, I'm thinking, how is it possible to treat more than 3 patients, basically? So is there a compassionate use program that could be working after the same model that you're doing in Israel in the US? Maybe, I don't know, who's the best person of you 3 to answer that?
You.
Well, definitely there are known programs that called different ways, accelerated early access programs, compassionate use programs that could be available. There are certain, obviously, criteria to whether APBD or GHF-201 is eligible for such program. I think there are several attributes that needs to be taken into consideration before going ahead and establishing such program. I think that also Professor Lossos mentioned this, that the program that we were entitled to do was limited to 3 patients only. I think that on a broader way, one of the criteria for such program is that it will not, I would say, interfere with any initiation of a clinical trial, because whatever you collect, whatever data you can collect in such setting is not eligible for such data to be submitted and accepted by the authorities. So in any way, you will have to do a clinical trial. So if you will start focusing on compassionate use program, it might cause some delays in getting us to the point where we can start a clinical study and get the product approved to the whole community, which obviously this won't be never a solution for the whole community. A compassionate use program, it will always be limited. So as I mentioned, we are doing whatever we can in order to expedite the process, in order to reach out the market as soon as possible. We are aligned with the FDA regulations. We having an open door access to them to reach out if we have any questions. This is what we will try to do moving forward, and hopefully we'll get to the market as soon as possible.